Hi, good morning, everyone. I hope last night was restful to you, and we're really happy to see you this morning. Some of you were here at 6:45 a.m. in your T-shirts, so I apologize if I got you up really early in a T-shirt. So it is 6:45 this evening. If you have a black shirt, and I think I said red, and it might be black, so whatever color you're in, either black or red. If you'll be here tonight at 6:45 p.m., then that's when the photo will take place. So that's T-shirt day. The first Lighthouse session that begins today at 11:15, the room has changed. So now it's in Congressional. So please think about that Lighthouse series. And again, the room is Congressional. So at the very end of today's meeting, because we're all going to gather again next year, we're going to talk about the where. And I bet that you can't guess where we're going to go. So we're going to tell you later this evening so you can get signed up. All right. Our first keynote speaker of this morning is Kevin Flanigan. I've known Kevin for about 20 or so years. I think we started together when we were very young. And he's really going to talk about his experience in the clinic and what this field looks like. So for now, it's Kevin.
Thank you, Pat. It's really a great honor to be asked to give the keynote speech, and Pat gave me sort of carte blanche. She suggested this title. So a little heads up, sometimes I like to do a lot of science stuff or a lot of things related to a single clinical trial. I don't intend to do that today. I want to take a sort of broader overview, if that's all right. And I do have some disclosures from companies that I've done work with or been funded by. The only mention I'm going to make of them in this trial is to put it in in this talk is put it in a little kind of bigger context, if you will. So today I want to do a brief reflection. This is where Pat made a mistake by saying I could reflect a little bit on where we've come from in research and care, and also give some thoughts that I think of every time when I come to the meeting and meet so many wonderful parents and families. Just some comments on a general philosophy, at least of the way I view it for entering this journey. A reminder then that we've made some great advances but not reached our goal, and there's promising work to do, and some lessons learned by at least one clinician scientist over the years. And just for fun, I want to start with this picture of Dr. Duchenne. I don't know if everybody's seen this picture, but Dr. Duchenne in the old days used to wander from one hospital to the other in Paris with his phoratic stimulator, where he'd give electrical stimulation to the nerves of the face. And he quite famously had a book about localization in neurology based on this work. Clearly, this was the days before institutional review boards and informed consent, but that was the case. But it was in one of his editions of this famous book that he first described what's clearly recognizable as a case of Duchenne muscular dystrophy. He called it something else, but he refers to it, but it's the disease that now holds his name. Actually, underneath that, I put another paper. I know I've talked to a lot of people who are kind of interested in the history of Duchenne dystrophy and so on. There's a wonderful review paper from 2003, going back and looking at all the references that predated Duchenne. So Dr. Marion in England and Dr. Conte in Italy and other references, if you like that sort of deep dive. But it's Duchenne that this is named after, of course. And between his description of it and about 1974, there really wasn't much change in what we did about the disease or how we treated it. This is a photo of Dr. Dan Jackman, who just passed away a couple of years ago at the age of 90. And he had retired at 89 from seeing patients at Johns Hopkins. And when I trained, I did my neurology residency and fellowship with him. And although he was famous for his work in myasthenia gravis, it's actually his work that has led to the mainstay of our treatment, glucocorticoids. So the very first paper in the English language was in the Lancet journal, very distinguished journal called Lancet, where he looked at 14 patients and treated them in an open label way with steroids. And this is a super interesting paper because, to me, it's super interesting because, number one, it shows what you could get into Lancet in those days. You could do an observational study, but it's a really classic paper of clinical observation, a hypothesis testing it in patients. And he closes with the statement that this observation of that there's significant side effects, but the presumed benefit would require a randomized trial. That randomized trial happened about 15 years later. It took till that was done in 1989 by Mendel and Griggs and others. So even when I trained there at Hopkins, there was some reluctance to use steroids, but that's all we've had for a very long time, although the benefit of it has been shown repeatedly to improve ambulation and outcome measures in boys. So that's the number one tool in our arsenal, steroids. The number two tool in our arsenal has remained attention to ventilatory support. There's many papers on this and experts here, Dr. Schell and others who are experts on this, but I want to point out that just nocturnal ventilation, this is a graph from a group at Newcastle University, where the care for boys with muscular dystrophy didn't really change, except about halfway through the period they studied, the group started doing nocturnal ventilatory support. Now we use BiPAP routinely, but also added spinal surgery for those boys who had quite progressive problems with scoliosis. And that's the curve on the right, and we can see significant differences in survival by using these two modalities. So that's, to me, the second and third pillar of the tools that we have in the pre-molecular therapeutic era. But it is the era of molecular therapeutics that we're in now, and because reflection is in the title of the talk, I will add that this is where, right around the time that I became involved with the field, with patients, with approaches to it. And I'm still kind of interested. I'd like to tell the story of this a little bit to point out that my actual entry into it came out of through the observation of someone walking into our clinic in early 2000 in Salt Lake City, Utah, where I was at the time, with this paper, and saying that in this paper, it showed that gentamicin could restore dystrophin function in the skeletal muscles of the MDX mouse. And the important thing to know here is that the MDX mouse has a non-sensor stop codon. Gentamicin is an antibiotic we use in the hospital all the time. It has, what it does is it gum up, if you will, the translational machinery of cells. It gums up bacterial machinery much worse than it does mammalian machinery, the ribosome, and it allows a bit of read-through of that stop codon to make low levels of dystrophin expression. So this was exciting. This was the first prospect, really, of restoring widespread systemic dystrophin expression. Now, the problem at the time, there's problems with use of this. I'll point out that it's not a practical way to approach this. Second is it doesn't work that well. But at the time, the real problem was there was no way to find these patients. We didn't know how to diagnose patients in 1999. 1999, diagnostic methods that were in common use only detected about 65% of the patient of mutations. Those were deletion mutations. And among those deletion mutations, it didn't even define the full extent of those. So that's where things were. So to find out and study patients, if gentamicin could induce these, how were we able to do it? And that's really where my interest from moving from the clinic into really thinking about these things came from. And what we did with my colleague at Bob Weiss at the University of Utah was to develop the first economical and sort of feasible, clinically feasible way. It was never commercialized, but clinically feasible way to do mutation testing to sequence the entire gene. So this methodology turned out to be important for a couple of reasons. Actually, the first thing I'll say is we were inundated with requests from across the country to make diagnoses in those 35% of boys whose diagnoses couldn't be made by the standard methodology. So we quickly realized we should make use of all these requests and this information that came in. And one of the things we did was to form a large multi-center network called the United Dystrophinopathy Project. And we maintain to this day this database. And I know many people here, their families are involved with this. You've given us clinical information, you've given us DNA to do analysis of. Now we have over 1,650 patients with Duchenne muscular dystrophy, Becker muscular dystrophy, or intermediate phenotypes. We may remain funded by the NIH to do this. And we continue to study the relationship between genotype and phenotype. I won't go into too many details about that here, except to say unusual mutations continue to tell us a lot about the organization of the dystrophin gene, the function of the dystrophin protein, and potential therapeutic approaches to get to better outcomes, better therapeutic dystrophins. And now we're also looking for genetic modifiers of disease. So that is, we know boys who make no dystrophin whatsoever. We know that some of them stop walking at 9 years old, and some of them stop walking at 13 years old. What's the difference in that curve? What's the difference from one end to the other? And we've used this database to define a couple of other modifiers, nowhere related to the X chromosome, to the DMD locus, other modifiers that have impacts on fibrosis or regeneration, that it may have an impact on disease. But at the time, the biggest contribution perhaps we made for this was to identify patients for trials of nonsense mutations. So this is sort of ancient history now in some way, not entirely, but I want to point out the first of these trials was of a drug called adalurin. Adalurin is an orally available, delivered compound that similarly causes stop codon read-through in experimental models, similar to how gentamicin does it. Not exactly the same mechanism, but very similar to it. And I always like to highlight this trial for a couple of reasons. It was 38 boys treated for 28 days with 1 of 3 doses in the very first paper. And, you know, we don't have approval of adalurin right now, but I think that this is a kind of key period. This was the first trial at systemic delivery to restore dystrophin throughout the body. And it was the first trial that really, in my mind, really started the discussion of proper outcome measures for DMD trials. The steroid trials that had come before that showed positive responses for things like muscle strength in quantitative muscle testing over time, but that doesn't really reflect function. We can't really say, what does a 5-pound increase in elbow flexion mean for you living your life? And there are plenty of talks about outcome measures, and you'll hear lots of discussions of things like the North Star Ambulatory Assessment, and we can discuss why the 6-minute walk test is not necessarily the best trial outcome to use, but this was the first trial that really approached it to say what could potentially lead to approval. How should we be studying these? How do we compare these to natural history groups? And in fact, I want to point out that lots of trials over the years have contributed to our understanding and our kind of future successes, the ongoing successes and future successes. I listed just 3 here of interest, each of which have a negative outcome. We should never say the term, as Eddie Smith pointed out to me last night, we should never say the term failed trials. We don't fail. We learn something from every one of these trials. And adalurin was an example. Drisapersen, the first, it was actually in the clinic slightly before eteplirsen, but not in the clinic, in trials, a different chemistry for exon skipping that did not meet criteria, but showed things about study design that were helpful. Mayo 029, it's a study that was done in Becker muscular dystrophy patients that showed some of the problems about using a heterogeneous population of patients that studied Becker and FSHD and limb girdle. All of these things are lessons kind of learned. The reason I want to highlight these, though, here is actually kind of a personal one. I'm here today, I've met 5 or 6 moms of family members who were in those very first trials, the adalurin trials. There are people here that I met 20 years ago at the University of Utah who came weekly for their infusions out there, or for their study they came out regularly for their study visits, brought their families out, invested all the time, effort, hope that all of us have for therapies. And I sometimes feel like we forget, we as clinicians, and I think even that we risk as people who have newly diagnosed children, realizing that the people around you here all have invested enormous effort to get us here where we are with new therapies, even though we haven't succeeded in showing all of these work. So I want to thank very much those parents who took part in these early trials personally, and I hope that if you get a chance to meet and talk to those people as well. So I want to talk a little bit about a general philosophy. And I'm sorry, those of you who visited me in clinic know that we have very long visits. I apologize. If you've ever come to the Nationwide Children's Clinic, we book 4-hour visits, and we sometimes run over because I yammer on too long in clinic. But part of my first visit is to really talk about this kind of philosophy, and I encourage all of you to think about these things that I might offer. I want to begin by saying there's not many things that are have-to items in this. I think it's sometimes easy to run into situations where people say, well, oh my gosh, you have to be doing that, and why aren't you doing that? And how come you're not doing that for your child? There are not many have-to items, I think, for parents in seeking care. I think the first one is to find a provider or providers that you trust that will take the time to explain and answer questions. Learn about those things they don't know about if you bring up questions about it. That's the key thing. We are a certified care center. I'm proud that we are. I think it's a great program. I know that not everybody can get to these centers where we have all this expertise in one place. And often it's parents who are educating primary pediatricians or others about the disease, bringing them papers, things they want to know about it. The important thing is finding people you trust to give you good advice who will help you make decisions yourself. So I want to emphasize that. Second is to find a steroid regimen that works for you and your family, or now vamorolone, I'll add as well. So the key, if you gave me one takeaway about steroids over 20 plus years looking at them, is the biggest distinction is being on some steroids or no steroids, or being, now I'm going to lump vamorolone in with that as well, being on it or not being on it. I do believe there may be differences in being in the regimens. The data shows differences in whether you're on, the intermittent 10 days on, 10 days off doesn't do as well as daily prednisone. Weekend dosing of prednisone and deflazacort, all of these things are shown to be roughly similar when we look at the long-term effects for them. So you have to find the right thing that works for your family. And I don't want anybody to feel if they can't get through insurance problems or access or something, one of these particular ones, the important thing is being on it. And we can say despite all of the side effects associated with steroids, we still see these long-term differences in boys who are on them or off them. Third thing is I would make sure that you have a good understanding of your son's mutation and its implications for therapeutics. So, it's sort of rough to get a crash course in biology when you get a new diagnosis like this with your son. I know that everybody here goes to providers at certified care centers or has access to the counseling, the genetic counseling through PPMD, has the opportunity to gain an understanding on this. Some of us, if you've not had your high school biology for a while, sometimes it takes a couple iterations, that's fine. Sometimes you come to a clinic like ours and God forbid the whiteboard is empty there, or we'll spend 10 minutes drawing diagrams about things like that. But it's important to have it, and it's important to have it because there are more therapeutic choices coming out, and some of those therapeutic choices that you'll be asked to make are dependent on understanding the mutation. It's important not to get too hung up on it, because functionally, if you don't make dystrophin, you don't make dystrophin. You have to treat the symptoms of it the same, although now in molecular therapies there are choices, and it's important to understand this. And the fourth thing is I would really carefully consider potential benefits and risks from new therapies. I share the excitement of everyone here about recent expansions of approvals and upcoming new therapies and new exciting things that might come down. It's really important when you look at those to say, what is the benefit and risk ratio for your son in particular? And that's one of our challenges as a field, to really understand fully the risk profile. Sometimes understanding that risk profile is not easy, digging that out of the materials that companies might have, for example, for us. And my fifth point is not here. My fifth point is avoid baloney. So, I was thinking back to 20 years of coming to this meeting. I saw it in Pittsburgh years ago. There have been things that I can only describe as fads that sometimes come through the community. Gosh, this e-stim machine or this therapy or this thing must be good. I heard it on Facebook. They're promising me if I come to the Dominican Republic with a bag full of cash, I can get these stem cells and so on. All of these things are out there. So I would encourage two things. I'm a big fan of the 1995 book by Carl Sagan, the great scientist and popularizer of science, called The Demon-Haunted World, Science is a Candle in the Dark. It's like a kind of Bible of rational thinking, and in there is a chapter about baloney detection, how to detect baloney. So I would encourage you, if you don't get the book, Michael Shermer, who used to write for Scientific American for many years. You can see online his synopsis of that chapter, the baloney detection kit. And I encourage you to keep that in mind as people come to you, well-meaning people from families, friends come and say, hey, I heard about this. If you pay this money, I think it'll fix your son. Keep those things in mind. I do want to talk a little bit about some of the new things and illustrate, for example, this idea of knowing what the mutation is and what it does. We're all excited about the microdystrophin approval, and I want to point out that microdystrophin, the concept of it derived from observations like this one from the laboratory of Kay Davies, where a large in-frame deletion in the central rod domain of the gene encompassing exons 17 through 48 results in the loss of about 46% of the protein, but walking at the time of the report to age 61, so really quite beneficial. This is the fundamental observation that comes from this. And you know, the work done by Scott Harper and Jeff Chamberlain, really, Scott Harper is a faculty member at our institution. I always like to highlight his work. This is from his thesis work in later publication, where he showed many different versions of microdystrophin proteins and measured their effects functionally in animals. And one of them there marked as R4, R23 is the protein version, that's the deletion that's encompassed in the current Sarepta approvals, which we're all aware of. And I won't dwell on very much here to say, I can tell you as a clinician, we're in a point where the somewhat surprising expansion of the approval, I think it's fair to say many of us were surprised by the breadth of that expansion of the approval, really raises for us some challenges in making sure how quickly we can roll these out to the population, how we can do so safely. For example, at our clinic, we do a lot of gene therapy. In our institution, we do a lot of gene therapy. And I just want to share with you as parents, we have parents who've been calling us for the past week or more, saying, you know, hey, can I come in next week and get my gene therapy? And it's obviously much more complicated than that, just to make sure we can do it safely and monitor patients. So I urge you to have a little bit of patience with your clinic as we think about these. But obviously, this is a wonderful milestone for the community. And I won't show tons of the data. You've seen some of the data showing benefits in North Star Ambulatory Assessment, time to stand, and other issues. This is from the publication in Annals of Neurology last year. The data in its entirety, I'll point out to you, is actually available online in memos from the FDA, which you can find without much difficulty. But I want to point out there are still other gene therapies being tried. So there are, in fact, 5 different gene therapies. My cartoon here is a little bit out of date with some of the details about the AAV serotype for a solids version, but all of them differ a little bit in what they do or don't include in the expressed transgene. So I'm not sure of where we'll be in compared. We're never going to have trials directly comparing these, I'm afraid. But I would encourage you to know that there are other choices as well coming down the pike that are worth studying, probably because some of them have differences, slight differences in what's specific to each of those gene therapy products. I won't belabor that. There's a session today from each of the gene therapy companies that I'd encourage you to go to look at those differences. But I want to put in a plug for my argument that we should be thinking about what a specific mutation is, particularly when it comes to exon skipping therapies. I think everyone here knows that exon skipping therapies are approaches to masking an exon in the splicing phase, so it's excluded from the final blueprint, the final messenger RNA. And there are, in fact, 4 drugs currently approved that are out there using it. Why would we care about these? Well, I'll point out that there are second-generation drugs that are much more efficient than these that are in trials right now from a lot of companies, many of which will be represented in another session today on dystrophin restoration therapy, so I encourage you to go there. But each of those has the possibility of making a product, a protein product, that is potentially better than what's expressed from microdystrophin therapies. So I would encourage you to keep that in mind. I'll show you what I mean as an example. We published, with the help of PPMD, their database, our database, French database, Italian database. We went through and looked at all of the Becker. We looked at all the patients with dystrophinopathies born with mutations that were the equivalent of exon 51 skipping. And when we look at these, we can see that some of these patients do very well. The light blue marks asymptomatic, including no elevation of CK. The pale green marks just hyper-CKemia, just elevations of CK as the symptoms with no weakness. The dark blue marks Becker muscular dystrophy. And it's fairly obvious that some of these mutations can be expected to have really excellent outcomes, as observed over the life of patients by Becker. That's what we're lacking in the microdystrophins, what we're very hopeful about, that the engineered microdystrophins are as good or better than these. But we have the natural experiment from these. From the Becker patients. So if you have a mutation amenable to exon 51 skipping, I'd urge you to still consider, for example, taking part in one of the trials of exon skipping therapies, or 44 skipping, or 45 skipping. There's ones coming down the road for many of these that really appear to be much more efficient. I also want to point out, for certain mutations, there's the prospect of something really good, and that's getting back to full-length dystrophin. And this is the only real data and gels and slides I'm going to present in the whole thing, blots here, so I'm sorry from the back. It doesn't show up very well. I'll tell you what this is. We actually, with the help of Sarepta, was gracious enough to give us drug for 3 patients. We looked at patients who had a single exon duplication. So if we have a single exon duplication and we skip that exon, we make full-length normal dystrophin. And this is a very messy slide. We have 3 boys dependent on their genotype, not on how severe they were. And we looked at the dystrophin expression as marked by the graphs. I'll just steer you to the bar graphs in the center. Those reflect the amount of dystrophin after treatment. The muddy-looking Western blots, the 4th panel down, I don't have a good pointer here, but those show that we can induce wild-type dystrophin expression over time. Well, what does that mean? I want to show you something from our paper that I want to say for what it is. It's a single patient in an open-label trial where we have all of the hopes of things going well, but it raises the possibility that restoring a highly functional dystrophin, like a full-length dystrophin, can have significant benefit or meaningful benefit. So this is our 18-year-old patient who mom came in about 18 months after he stayed on the drug now. Mom came in and said, he can take his shirt back off again, and he can reach down to pick his drink up off the floor. He can sit up in bed, and he couldn't do that before. Now, for every parent, everybody with muscular dystrophy in this room, you know that this is not normal function. But in my years in the field, I've never heard someone come in and say they've gained a function like this back. So to me, it raises a great deal of optimism that even in a severely affected state, if we can restore highly functional protein at meaningful levels, and those levels are only about 8% in that boy, we might be able to make some differences. So we'll see if this continues to increase over time. So I'm going to close with what have I learned. You've got to forgive me. Blame Pat, because she said I can do this reflection thing. So this is But what I learned is nothing about science and nothing about medicine. It's from this meeting, from coming to this meeting for 20 years and seeing patients in clinic. What I've learned is something about family. I come from a big Irish Catholic family. I thought I kind of understood family. What I learned in clinic is families come in all sorts of shapes and sizes. You can choose your family. Families are those who really take care of each other through through challenges and troubles. And I'm humbled every week in clinic to see how families come together and how they define themselves and as a person around this devastating or challenging disease. I want to say that. And I know that sounds a little trite, but it's the reason we bring people from our clinic or from our laboratory to clinic to meet families, to understand why we're doing what we do all the time in the laboratory. And I'll say love, too. I am always just stunned by the amount of love that I see demonstrated right in our clinic room. That little 8x8 clinic room, sterile hospital thing, it's something we really, that I've learned personally more about from being able to be part of this community. Quality of life. One of the first things we teach residents when they come to our clinic is, don't throw out your assumptions of quality of life. What you think you're looking at doesn't mean anything. You need to ask a family. You need to ask a boy, how are you doing? What are you doing? What brings you joy? What brings you happiness? And residents who train with us, just as I had to learn, any assumptions I have about it are meaningless compared to what we see from patients in clinic. I want to add generosity, that generosity of spirit from each of you. I'm just amazed all the time at how people affected by this disease who've been through this journey together, been through this journey themselves, are more than happy to share their expertise, to share everything they've learned, to really help to ease the burden for somebody else. And I think that generosity is incredible, and I've learned from it. And community as well. It's kind of amazing when I think back to the first meetings I went to in Pittsburgh years ago, how this community has changed, how this community has grown, how people have a sense of belonging and get really great information from a meeting like this. And then finally, I'll say hope. You know, years ago, when I first started seeing patients, I couldn't look any parent in the eye and say, I know there's something either available today or on the cusp or being tested that I know really has the chance of making a meaningful impact on your family's life, on your boy's life. We're not there now. We have a lot of hope, not just from what's been approved, but lots of things. You're going to hear sessions this afternoon from great scientists, from great biotech partners, from great people working on many aspects of this, and I think the world is really changing for this fairly quickly. And I think the story for the young men diagnosed, the boys diagnosed today, is really a different one from the ones diagnosed in Dr. Drachman's time. So with that, I'm going to close, and I just need to put up a thank you to our laboratory and many people in our center who are involved in patient care, and this is kind of a laboratory-based one. I apologize. I've left off a few colleagues, Ann Conley and other members of the clinical staff. I pulled it from a lab-based one. I do want to thank particularly the Parent Project Muscular Dystrophy and share one thing. I don't know if you guys know, one of the things that Parent Project Muscular Dystrophy has done for many years now is to support an annual course that we teach at Nationwide Children's Hospital, where we bring in 60 young trainees per year, and for a week they have an incredible experience in learning about muscle disease, learning about clinical care, meeting with parents, seeing patients, and the goal is to train the next generation of young investigators, young clinicians. And it gives me it's really gratifying to come to this meeting or other meetings or scientific meetings and see all these people who are in the field who are studying muscular dystrophy. And it's really due to the support of Parent Project Muscular Dystrophy. So with that, I'll close. Thank you very much.